Welcome back. Here we are in Chapter 20, Part 2, where we are continuing our discussion on specific tools for manipulating DNA and biotechnology. Our next tool is known as PCR and has had huge ramifications in biotechnology in terms of forensics and crime. You've heard about PCR for COVID and other things. It looks complicated, but it's actually not too bad as soon as you understand the pieces and the concepts behind which drive PCR. So PCR is a way of amplifying specific sequences of DNA. And the ingredients is that you have a template, an original sample of DNA. It could be from cells or so forth. You add some primers that flank the 3' and 5' ends of the sequence that you're interested in. So you have some primers and you also have nucleotides. That's almost all that you need for this 3-step process. The only other thing that you will need is an enzyme from an organism that lives in a hot spring, and it's a DNA polymerase that originates. There are two major forms of it that we've isolated from bacteria that were found in hot springs, and so they can survive the heating part. So step one is denaturation, where we heat this thing up to almost boiling, and that's why you have to have an enzyme that can withstand that. So we have an enzyme from an organism that lives in a very, very thermogenic environment. That denatures the DNA. And then we have a rapid cooling. That's called annealing. And what happens is when you cool rapidly, yes, these two sequences could come back together, but these primers will actually come back in quicker and prevent the original sequences in many cases from re-annealing completely. And then what you do is you get to the right temperature and you have those nucleotides in there, and the DNA polymerase will then use that primer to create a final strand of DNA. It's just that simple. Now there's a little more dynamics to it to explain how we end up amplifying this specific sequence, and we're going to show you that. So here is the scientist who discovered the concept of PCR. And the key was that you needed a heat-stable DNA polymerase. So the story of how Kary Mullis developed PCR is actually really, really cool. And I'm going to give you the article that has the story about how he figured PCR out. The original PCR, we used Thermus aquaticus, which you can find it lives in Yellowstone National Park. In the hot springs in Yellowstone National Park, the DNA polymerase from that Thermus aquaticus was what Kary originally used. So you need that TAC is how we call it, TAC polymerase. PFU, I'll talk about in a little bit. That's one that's actually got a little bit better fidelity. You need primers, and you need that flank the sequence that you're interested in, and then you need the original DNA. The problem with TAC is that it is a bacterial one. So is PFU, but PFU has a proofreading function that TAC doesn't. So occasionally you get some errors in there, being a bacterial DNA polymerase. So you really can't necessarily clone by itself. It is a part of the overall cloning process, and you can do it, but you have to check for errors. Like I said in the previous slide, TAC polymerase was identified originally from the Thermus aquaticus in Yellowstone. And this is a picture I took a few years ago, Grand Prismatic Spring, where Thermus aquaticus does live. This is the site right here where the PFU polymerase was identified. It was on a beach in Italy near some volcanic activity in the northern part of Italy, kind of near Milan. Just found it on a beach. It's pretty neat. That's the beauty of some of these things, is that if you have just a little bit of understanding, you can use your creativity to come up with new ways of going about things. So the technique is that you have some genomic DNA, which has your target sequence of interest. You add the primers, which will flank one end and the other end of your target sequence. So you have to have primers that will prime on one strand and primers that will prime on the other, and they flank the ends of the sequence of interest. Then what you do is you denature the DNA, you anneal with the primers, and then you bring the temperature back up to the ideal temperature for your polymerase. The nucleotides are there, and they will synthesize off of those primers. What happens is, yes, if you have a long extension here, you will get long products, but those then become templates for the shorter ones as you go. And as you do multiple cycles of this, you don't ever do, or you very rarely do just one cycle of this. You might do as many as 30 cycles, and by the time you get to cycle 30, the shorter sequence products are so much more abundant that you can run gel electrophoresis and isolate just those intermediate products that have your sequence of interest rather than the long ones that may have originated in the early stages. So we amplify it a few times, usually about 20 to 30 cycles, and then we purify for the intermediate length products that we're interested in. So here's the basic setup. You have your primers that flank the sequence of interest. They can be in the sequence or they can be a little bit beyond the sequence if you need it. You can actually modify the primers a little bit. I've put these little ends on here in order to say add restriction endonuclease ends there so that you end up with something that you can then digest and then use to insert as a clone in one of your plasmid vectors. So you could just amplify the sequence of interest, or you can amplify the sequence with slight modifications at the ends, either to create markers that you can detect, or to, again, digest with a restriction enzyme so that you can then clone it into an expression vector of some kind. Here's another video again for you. Please download the PowerPoint and watch this video. It's an excellent explanation of how PCR, the PCR reaction works and why we end up amplifying just the short sequence and not amplifying long extended sequences with this type of technique. And here's another video, though this one is really interesting because what we're showing you here is is not only the antibiotic resistance, but if you see this gray gene right here, this gray gene is actually called LAC-Z. And that allows this plasmid to express a protein that when lactose is present in the growth media, will cause the bacterial colony to be blue. So what this is showing you is that you can clip a piece of DNA out of here and you can amplify it and do that sort of a thing. Drop it in, and when it's dropped in successfully, it will be resistant to the ampicillin, but will not be blue. So it's going to talk about using a colorimetric marker in addition to an antibiotic marker so that we know that we have the human DNA cloned into this plasmid. And so PCR is a massively important technique. We can use it for all sorts of things, for archaeology. Like, for instance, we can compare the DNA from a woolly mammoth that lived 40,000 years ago to modern elephants. DNA is very, very durable substance in many cases. It's not perfect, but certainly can survive being frozen for 40,000 years, and you can look at the sequences and compare them to modern elephants. You can identify from very, very small amounts of DNA, like from a fingerprint, or from tiny amounts of evidence found at a crime scene. You can clone from DNA from single cells or from small populations of cells. Like, for instance, we can detect virus even before the disorder has manifested as an actual viral infection, et cetera, et cetera. So very, very powerful technique when used properly. It is possible to have errors. PCR is a very sensitive technique, so if you don't have the right primers or other things, you get contamination. You can get some false things. There are some drawbacks, but overall, the positives far outweigh the negatives. Here's sort of an example of use of PCR for understanding what a cell is expressing in that cell. We talk about DNA as genomic DNA. Well, we can create a molecule called cDNA. What cDNA is it is synthesized using reverse transcriptase, which we originally identified in viruses like HIV. HIV is an RNA virus, and when it gets inserted into a target cell, it will turn that RNA back into DNA. And so we can use that enzyme and we can isolate mRNAs from a cell or tissues. We can turn it into cDNA and then amplify it using PCR, and then looking at the results on gel electrophoresis from different samples that we have and comparing the amounts of DNA that we have. So there's lots of possibilities here. And here's an example of this technology. It's called a cDNA microarray, where what you do is you selectively amplify, or you create cDNA with a special red marker from tissue 1. So the cDNA has a red fluorophore on the cDNA, and you can take another tissue and use a green fluorophore for that. And then what you do is if you see genes expressed in one but not the other, you'll see one color. For instance, this dot right here is red because only the red gene is significantly, the gene from tissue 1 expresses that gene, but tissue 2 doesn't. Whereas if you have a green dot, tissue 2 is expressing it, but tissue 1 isn't. If you have a yellow dot, that's where when green and yellow light combines, you get, or green and red light combine, you get a yellow color. And so yellow are the genes where you see both tissues. And if it's dark, then you don't have expression in either of those tissues. This is an example of a colorimetric cDNA microarray, and you can do this for literally thousands or tens of thousands of genes. This is a bit of a repeat figure. It's a question asking in your book, asking about your concept understanding. I strongly encourage you to go through those questions while you go through the reading to make sure you understand. Mastering is along those lines as well. What we have is our vector. This is a cloning vector. Sometimes they're cloning for growing up additional copies, and sometimes we have expression vectors, which are designed to express in bacteria or express in eukaryotic. You get those DNA fragments maybe from PCR or maybe from some other source like a restriction enzyme cut. And then you can drop those in not only in a specific site, but also in a specific orientation. Our final section we're going to talk about involves editing genes and genomes. There are multiple techniques for doing this. You can do things in vitro where you can introduce mutations. You can do chemical mutagenesis. You can do site-directed mutagenesis. And we're going to talk about the technique called CRISPR-Cas9. When we introduce a mutation, maybe we change the function, or maybe we destroy the function. Very, very, very rarely we can cause, when I say change, we can also induce function. That's extremely rare. It is possible. When we return that gene to the cell in whatever form it is, we can then examine the effect on phenotype or use it for therapy in some cases. So with CRISPR-Cas9, it's going to be really difficult to go through all of the pieces. It goes a little bit above and beyond what we're going to be capable of, at least at this point with your level of expertise. But understand that there are basically three pieces three things that go into it. First of all, you have the original DNA of your target. And that could be isolated DNA, it could be DNA inside a cell, or even inside an intact organism. It's actually that powerful. You have the Cas9 protein. This is a combination of exonuclease type of it's a targeted type of exonuclease. It can cut double-stranded at very specific locations, but it's a variable cutter. Whereas restriction enzymes are cut at a single highly specific spot, Cas9 will also cut at a highly specific site, but it can be varied using this guide RNA, this single-stranded guide RNA, and you can change that RNA and make it so that it will target almost any sequence in an intact cell or a tissue or in vitro. And using that guide RNA, that's the CRISPR. It stands for resistant, repeating sequence. It was originally found in bacteria that had genomes that where it had been these bacteria had been infected by viruses and had fought off the viruses, but retained some of the viral sequence so that if it was infected with the same virus again, it could attack it right away. That's the short story of this. That it maintained these viral sequences, not the entire virus, in order to better fend off a viral infection in the future. But with this guide RNA, that's the CRISPR, the Cas9 protein in your original sequence, you can insert mutations if you want. You can delete whole genes if you use a couple of different guide RNAs. Like, for instance, you can have one for one site and one for another site over here, and you can cause a deletion to occur. You can insert, depending on the configuration of the guide RNA, and modify. So I have a video for you on that so that you can see that a little easier. And as I said, you can insert mutations, you can cause deletions. This video I will make available for you on Brightspace. Please do watch it. Again, we're not getting after the exact perfect mechanism here. We're just getting after what this thing can do. So you should be able to recognize what is possible with the CRISPR-Cas9, what the major ingredients are. You have the Cas9 protein, you have the guide RNA, and you have your RNA target. And then what are the possibilities for what can be achieved? And what's really cool about the CRISPR-Cas9 system was that it was two very talented female scientists who pioneered and discovered the system and its adaptability for therapeutics and for gene editing. They both had different expertise. Jennifer Doudna was an expert in RNA, and Emmanuelle Charpentier, she was an expert in biochemistry and protein function and looking at resistance. And together they shared the Nobel Prize in 2020, which was astonishingly fast for the Nobels. Normally, people get Nobel Prizes 20 or 30 years after they make their discoveries. Jennifer and Emmanuelle got theirs like in less than 10. So that's just how impactful CRISPR-Cas9 has been already in our society. Therapies for things like sickle cell anemia and other autosomal recessive disorders, they're already pioneering CRISPR-Cas9 therapies for these disorders. Pretty cool stuff, and I'm going to have an article for you so you can kind of read, especially about Jennifer, since she's American. The American public, press follows her a little more closely, but it talks about them both, so it's a good article. And this concludes our part 2. So the idea that biotechnology is going to affect all sorts of aspects and already has. And this is one of the reasons why UIndy has put cell biology so early in the curriculum. As an undergraduate myself, I didn't take cell biology until my senior year and then followed up with cell biology in graduate school. We give you cell biology up front for the fact that so much of the biotechnology is already manifest in our society that you kind of have to have some of that cell biology just to kind of get going in the major areas of the other parts of our curriculum, like microbiology and genetics and advanced cell biology and cancer biology and therapeutics. You think about mRNA vaccines and again, gene therapy, GMOs. It's everywhere in our society. And the more you know, the stronger you'll be and the better you'll be able to make actual informed decisions rather than what somebody says in social media. So thank you for your time and good luck. And please hit me up with any questions that you might have. Talk to you soon.